Okay, well, I was asked to give a kind of general overview of the contribution that X-ray crystallography has made to our society in the 20th century, and maybe make some projections into the 21st century. And I will start off by pointing out that study of crystals is not just 100 years old. People have been mystified and enchanted by the beauty and symmetry of natural crystals throughout recorded history. This is not anything new. And indeed, we even know that on the planet Krypton, Superman's home planet, they were enchanted by crystals as well, and they chose to embed all the knowledge of their planet into crystals, something that we have yet to figure out how to do, but we're working on it very diligently. So this is a picture of the planet Krypton, Superman's home planet. And a lot of other people are enchanted by crystals as well. I picked this crystal awareness guide up some years ago in California, which does seem to have an attraction for people who feel that crystals will improve their life. They can rub them on their skin. It will improve their health. It will improve there's some words they use that I don't remember, chakra or karma or something like that. And the Legion of Light still exists, and they're still selling stuff on the internet. And if you Google Legion of Light, you will find they have all kinds of products they will be happy to sell you relating to the influence of crystals on our daily lives. So I'm not sure that I'm recommending this to any of you, but you might, out of curiosity, like to look it up. Anyhow, getting back to some real science. The 1914 Nobel Prize in Physics was awarded to Max von Laue. He did the original work to confirm the wave nature of X-rays. X-rays have been discovered some 10 years earlier, a little bit more than 10 years earlier, by Roentgen, and nobody was quite sure what they were, which is why they were called X-rays. And von Laue had the idea that if X-rays were in fact electromagnetic radiation and had the nature of waves, which is one of the proposals, that they should be diffracted by crystals. Now, being a good physicist, I believe he used the most expensive crystal he could find, which was gold, and he shot a narrow beam of X-rays into his very thin foil of gold, a photographic plate behind it, and indeed he obtained a diffraction pattern from the crystals. Representation of a typical diffraction pattern from a crystalline material illuminated by a beam of X-rays and detected on a photographic plate or a photographic film is shown on the right here. Now, von Laue was a theoretical physicist, and he basically lost interest in X-rays and diffraction at this point, and he went on to pursue a career in theoretical physics and relativity and particle physics and so on, and really played no further role in the development of the subject. However, a young man, a very good-looking, handsome young man called Lawrence Bragg, read von Laue's work. He was a 22-year-old, first-year graduate student at the University of Cambridge. He was a student of J.J. Thomson. Now, J.J. Thomson himself had won the 1906 Nobel Prize in Physics essentially for the discovery of the electron. And Bragg, having learned of von Laue's work, as he tells the story, was walking one day through the gardens of his college in Cambridge, and musing on this work, and he realized that X-rays could be used to give us an insight into the internal structure of crystalline materials. And he apparently sat down and worked on this. He developed a much, much easier method of analysis than von Laue had used. And he had the distinct advantage that his dad was a physics professor. His father, William Henry Bragg, was a professor of physics at the University of Leeds. And so one imagines that the young 22-year-old Lawrence Bragg goes up to his dad and says, "Hey Dad, I've had this great idea. Do you think you could help me build some stuff to confirm that it works?" So the two of them designed this amazing machine, which they called a diffractometer. And that would basically bombard a crystal with a very narrow, focused beam of X-rays and be able to detect whether or not a diffraction pattern was generated as a consequence of this illumination, and to quantitatively measure the position of those diffraction spots and make deductions about the crystal structure of the material that they were looking at. Now, I was fortunate enough, when I was a junior in high school, back in 1951, I was in high school in southeast London, in a suburb, and my school would get invitations to Christmas lectures at the Royal Institution in London. And they would typically send each school in the area 4 to 6 invitations that these young students could go up to the Royal Institution on Albemarle Street in London, a very old establishment. Sir Humphrey Davy was one of its first speakers some 200 years ago. Michael Faraday was another one of its distinguished members. Anyway, 1961, I attended a Christmas lecture at the Royal Institution. There was this really, really old man who gave the Christmas lecture to school children. And he was so old, I couldn't believe that a guy this old was still alive. As it happens, I'm now that age, as is Dr. Anderson. And I guess Dr. Moffat as well. Anyway, this old man was Lawrence Bragg. And he gave the Christmas lecture to school kids that I attended. And afterwards, we were invited to go up and talk to him, so I had an opportunity to have a conversation with Lawrence Bragg. And I remember absolutely nothing about this. Well, that's not quite true. I remember one thing about the entire activity. I remember this is nearly 60 years ago, okay? But one thing about this. This is London in December, and the weather is miserable. And he said, okay, I'm going to turn out the lights, and you all got umbrellas. I'm going to turn out the lights, I'm going to put on one very small, bright light, and I would like you to look at it through your umbrella. So everybody open your umbrella and look at this bright light, and you will see a diffraction pattern. And we did, and I remember that. Now, none of you have umbrellas with you today, and I don't have a really bright light that I can demonstrate this with, but we can kind of do the same thing these days, and Lawrence Bragg didn't have the benefit of a laser pointer like I do. But I brought with me a small piece of silk, which has a very fine array of fibers in it. I brought with me my laser pointer. Okay, so there's the red light. If I shine the laser pointer through the red light, I hope the camera is able to see this. There's the laser pointer. I put the silk in front of it, and lo and behold, we see the diffraction pattern from the fibers in the silk. And the mathematics that describes that pattern is exactly the same as the mathematics that describes a diffraction pattern of X-rays moving through a very thin crystal. And indeed, if you look at that pattern, and I'm not cheating, there's a single dot, I move the silk in front of it, we see multiple dots, which is the diffraction pattern. As I rotate the silk, the pattern will rotate. I have multiple thicknesses by folding the silk over. I get multiple, more spots present. Physics is exactly the same for doing that as it is for X-ray analysis. I learned this trick from Lawrence Bragg. Anyhow, moving along. Lawrence Bragg, incidentally, won the Nobel Prize in Physics at the age of 25. He's still the youngest ever Nobel Prize winner. Now, having established that their method worked, they went ahead and they started determining the crystal structures of a number of materials. Because they were in Cambridge, whose reputation was set by, I guess, Ernest Rutherford, we don't do any experiments that cost any money. They didn't have the National Science Foundation in those days to get money. They did their experiment not on gold like von Laue. They did their experiments on sodium chloride. Their first experiment. They determined that the first people to determine the crystal structure of sodium chloride and other salts related to sodium chloride. They looked at metals. They determined the crystal structure of copper. They did, in fact, determine the crystal structure of gold as being face-centered cubic. For some reason, they couldn't manage iron, and somebody else did the experiment to determine the crystal structure of iron. But their method turned out to be so powerful, it was very quickly adopted by a lot of other research workers. It led to the first measurements of bond lengths, which the chemists were very grateful for. It led to the concept of an atomic radius and an ionic radius, and they made the first determinations of atomic and ionic radii. Later on, Linus Pauling, yet another Nobel Prize winner, used a number of these values of ionic and atomic radii to come up with Pauling's rules for predicting crystal structures. Further down the road, people like Max Perutz and Dorothy Hodgkin used X-ray diffraction to determine crystal structures of biological molecules. Perutz with myoglobin. Hodgkin determined the crystal structure of penicillin and vitamin B12. The geologists got in on the job with the mineralogists, crystal structures of rocks and minerals, quartz and so on, olivine, gemstones, alloys, ceramic materials. All of these had their crystal structures determined by X-ray diffraction. Further along, identification of defects in crystal structures. These defects that affect things like the properties of the mechanical, electronic, and magnetic properties of useful engineering materials were identified using X-ray diffraction or electron diffraction. Turns out that on account of the wave nature of electrons and neutrons, and in fact all particles, beams of electrons and neutrons can be used to perform diffraction experiments on materials that may not be amenable to analysis by X-ray diffraction. Argonne, of course, for some time had a pulse neutron source that was used extensively for neutron diffraction experiments, and currently it has the APS, Advanced Photon Source, that you'll be hearing about from Professor Moffat in a few minutes. Pretty much any electron microscope these days can perform electron diffraction experiments. The physics of the analysis is almost identical to the method originated by the Braggs. The IC, International Center for Diffraction Data, currently in 2013 edition, currently holds structural data for 778,833 different crystalline structures. Just about every substance for which we know the molecular or crystal structure, that has been determined by a diffraction technique, mostly X-rays, some of them neutrons or diffraction or electron diffraction, using essentially techniques pioneered by the Braggs. So I'm going to talk about a few specific applications. Most of them will be from my own field, which is materials science, and I've chosen one particular application from biology. So we'll start off with materials application. High-strength alloys. Pure metals are remarkably weak. They're very weak on account of the presence in their crystal structure of a certain type of defect called a dislocation. Now this is a piece of very high purity aluminum wire. I hope the TV cameras are able to see this. It's very high purity aluminum wire. It's incredibly soft. Now, I'm not going to say this is as soft as butter, but it is about as soft as Parmesan cheese. Okay, it is no problem whatsoever to bend it into a circle, tie a knot in it, tie a square knot or whatever. Okay, I can just take this piece of wire, its diameter is about 1 millimeter, and I can trivially easily deform it. Pure metals are incredibly soft and weak. You certainly would not want to fly in a Boeing aircraft that had its wings made out of this stuff. Well, I wouldn't anyway, maybe other people would. Now, the reason that pure metals are incredibly weak, and it turns out that they're much weaker than the strength of the bonds between the atoms would suggest, is that they contain a certain type of defect called a dislocation. Now, another student of J.J. Thomson, G.I. Taylor, Geoffrey Taylor, Sir Geoffrey Taylor, I guess I should call him, came up with the explanation for the incredible weakness of pure metals, whether it's pure aluminum, pure gold, pure iron. They're all very, very soft and weak. And he suggested that it was due to the presence of a type of defect called a dislocation. Now, conventional alloying has been going on for something like 10,000 years. The earliest known bronze is an alloy of copper with something else. The earliest known bronzes date back to about 8 to 10,000 BC, according to the archaeologists. Now, we know that conventional alloy just introduces atoms, basically of different sizes, into the crystal structure of the parent material. Since these atoms are of different sizes, the tin in bronze is a different size than the copper atom, it basically introduces what you might call speed bumps into the crystal planes. These speed bumps hinder the motion of dislocations and make it more difficult to move them, which makes the material stronger. And Geoffrey Taylor's idea of what a dislocation might look like is shown on the top left of this picture. The first pictures of actual dislocations in actual materials and actual solids were made in silicon using a diffraction technique called X-ray topology. Now, this is not exactly Bragg's technique, but the fact that we get contrast that enables us to see these dislocations, these wiggly lines in the picture on the right, are actually images of crystal dislocations at a macroscopic scale. The fact that we can see them at all is due to the fact that the crystal planes on the around the dislocation, the crystal planes are bent and distorted, and consequently they diffract the X-rays in a different manner than the undistorted crystal away from the dislocation. And when we image it, the X-rays that are diffracted differently end up giving us a dark line on the picture, showing us where the dislocation in fact is. Now, subsequently, electron microscopy has done a lot more work on the structure and the presence of dislocations, and we know that Taylor's hypothesis is 100% correct. So, getting back to our topic of high-strength alloys and the contribution of X-ray diffraction. In 1906, a young German called Alfred Wilde accidentally discovered that an alloy of aluminum with copper developed a remarkably high strength when left to age for several days. Now, I say it was accidental. It truly was accidental. He was working for a research establishment in Germany, and he was preparing these alloys, which he assumed were going to be strengthened in the same way as alloys known since antiquity, like bronze and brass and so on. And he prepared a series of alloys. Now, in those days, they had a 6-day work week, and so he'd been working on a Saturday morning, and he prepared some alloys. He'd already tested some of them, but he hadn't finished the mechanical testing, the strength testing, by the time he wanted to go home, because apparently he was going to a dance that evening with his fiancée. And so he left a whole bunch of these alloys untested on his desk. And on the Monday morning, he came in and continued the testing, and lo and behold, these alloys that had been left over the weekend were way, way stronger than any other aluminum alloy that had ever been developed up until that time, and way stronger than the aluminum alloys, the same alloys that he tested on the Saturday morning. And so they didn't know what on earth was going on, and they assumed it was like people. You get better as you get older. Is that correct? That's absolutely correct. Right, Keith? That's correct. Yeah. Okay. And so they called it age hardening. Okay. The alloys are being aged like fine wine, or like fine people, or fine scotch. The alloys were getting better because they were being aged. Now they looked at these things in the microscope. In those days, all they had were optical microscopes. Nobody could see any difference. The before and the after looked exactly the same. Nobody could figure out what was going on. There are a lot of hypotheses, obviously. I'm talking about science here, like scientists like to submit hypotheses, particularly when there's no evidence to contradict their hypothesis. And so nobody knew what was going on. They put this stuff in production anyway. They called it duralumin. In fact, the alloy they call duralumin is now sold in this country pretty much under the name of 2024, an aluminum copper alloy that age hardens. That took until 1938, when Andre Guinier and George Preston, Andre Guinier in France and George Preston in England, independently related streaks that they saw in X-ray diffraction patterns of these alloys after aging. Now we can see the streaks here. These are the main diffraction spots from the X-ray analysis from the aluminum, and these streaks were not present before aging. But after aging, the X-ray diffraction pattern developed these streaks around the main diffraction spots, and their analysis indicated that these streaks were due to the development of copper enrichment on certain crystal planes, which nowadays we call GP zones, Guinier-Preston zones, okay? Andre Guinier, George Preston. We nowadays call these GP zones. And these zones, unlike the old, from antiquity alloying, where the alloying elements act like speed bumps, these enrichment zones are more like roadblocks. These are more like a semi that's overturned on the freeway and blocked all the lanes. They really, really do a first-class job of stopping these dislocations that Geoffrey Taylor had proposed. These zones essentially shut down that kind of dislocation motion very effectively, giving tremendous strength to these alloys. And most small light planes these days are still using these alloys that Wilhelm first empirically discovered 100 years ago. Things like Cessnas and Pipers and so on extensively use those alloys. Once the strengthening mechanism had been identified, it now allowed the engineers to move in and use the knowledge of what exactly was going on to engineer new alloys that were strengthened by the same mechanism. Now this, if you like, becomes the first known application of nano-engineering. Now we hear about nano-engineering, nanotechnology and so on a lot these days, but the first real application of nano-engineering dates back to the discovery by Guinier and Preston of these zones, and then applications by metallurgists to develop new alloys strengthening by the same mechanism. So I'm just going to give you a real quick run through of some of the alloys that have subsequently been developed based on this mechanism. The nickel alloys used for jet engine turbine blades. This is a very unpleasant environment. Jet engine turbine blades spinning, very high speed, many tens of thousands of RPM typically, so they're loaded by very high centripetal forces. They are running at very high temperatures in a very unpleasant oxidizing environment. And in a typical modern airliner, these turbine blades are expected to last somewhere between 5,000 and 10,000 hours of operation before being replaced in overhaul. And these alloys are strengthened by exactly the mechanism that Grenier and Preston first deduced using X-ray diffraction techniques. The boundaries between individual crystals in these alloys. There's currently a lot of research work going on looking at the boundaries between one individual crystallite and its neighbor. Because these boundaries also, at high temperatures, act as points of weakness. The crystals can slide past each other in a process known as grain boundary sliding. It turns out that a certain orientation relationship known as a sigma-3 boundary is extremely resistant to this grain boundary sliding. That is an active area of research in universities and in the companies like Pratt & Whitney, Rolls-Royce, and so on, who manufacture jet engines. And these orientation relationships between one grain, one crystallite, and its neighboring crystallite along the boundary, these orientation relationships are currently being investigated using electron diffraction in electron microscopes. Compressors of jet engines. These don't have to withstand high temperatures, but these fan blades certainly have to withstand very high stresses. They're for the most part these days made out of titanium alloys. The titanium alloys that are used are strengthened by this process of developing these zones enriched in one alloying element that act as these very effective obstacles to the movement of dislocations. The very lightweight aluminum alloys used for airframes. The original alloy developed by Wilm back in 1906 is kind of obsolete for airliners. This will be the wing of a modern airline. Nowadays they're using aluminum lithium alloys, which have a very low density, they're very lightweight alloys, and they're also extremely strong on account of the strengthening mechanism that was first identified using X-ray diffraction. Okay, well, my one example of biology I've stolen from Professor Moffat, but he said he wasn't going to talk about it anyway, so I thought I would. The iconic image, maybe of our time, maybe of the 20th century, is the X-ray diffraction image of DNA that we'll be looking at in a moment. By 1950, it had been fairly well established by chemists such as Alexander Todd and Linus Pauling that DNA molecules contain a ribose sugar and 4 types of organic bases, which every grade school student these days knows as by the initials of A, C, G, and T. And Maurice Wilkins, who at that time was at King's College London, proposed, following along ideas that Max Perutz had developed for identifying the structure of myoglobin, that maybe X-ray diffraction would be a very handy tool to investigate the structure of DNA. Unfortunately, DNA turned out to be very difficult to make a crystal. And in order to do X-ray crystallography, you need a crystal, or at least a pseudo-crystal. Wilkins, after some effort, was able to prepare fibers of semi-crystalline DNA. I believe he used DNA from the thymus of a calf. And this is a picture of a typical fiber. These fibers were very easily damaged by the intensity of the X-ray beam. The X-ray beam would deposit a lot of energy into the fiber, would tend to burn holes in it. So one of the experts at King's College London, Rosalind Franklin, developed a technique for protecting the fibers from damage, essentially by having the fiber enveloped in a wet hydrogen atmosphere during the experiment. And she developed the specialized diffractometer for doing this. Now, I'm not a biologist, and I'm sure that Professor Moffat is going to contradict everything I'm telling you about this in a few minutes, but you're going to get my version of the story now. Anyway, so Rosalind Franklin developed a technique for doing the X-ray diffraction of these fibers. And the iconic photograph of their research was the so-called Photo 51. If you just type into Google Photo 51, you'll come up with this picture. This is X-ray diffraction pattern. The experiment was actually done by one of Franklin's students, Raymond Gosling, and this is the X-ray diffraction photograph that he took exposed on regular photographic film. And you'll notice it shows spots. Well, they're rather diffuse because these crystals were not perfect crystals. They were kind of pseudo-crystals. You'll see a bunch of spots in the shape of a cross. You'll see a big exposed region at the top and the bottom, a bunch of smaller spots here. You'll see an obvious gap in the pattern of spots on that X at these points. The X shape, we now know, is a consequence of the molecule being helical in shape. By measuring the distance between these very intense spots at the top and the bottom, we're able to determine that there's a spacing in the crystal, a significant spacing in the crystal, that is 0.34 nanometers. That turns out to be the spacing between what we now know as the base pairs in the DNA molecule. And the lesser spots down here, closer together, these essentially correspond to the pitch of the helix, and there are 10 of these spots between each of these big spots, telling us that the base pair, that the helix itself had a pitch of 3.4 nanometers, which is 10 times the spacing between the base pairs. In other words, there were 10 base pairs for each turn of the helix. The missing spot at the fourth position, okay, counting out from the center, 1, 2, 3, and the fourth one is missing. The missing spot, I say it's missing, you can imagine there might be one there, there certainly isn't one there. The missing spot at the fourth position told Crick, who saw this picture because James Watson had seen it and told him what the missing spot at the fourth position told Crick, who was a crystallographer, that the helix, in fact, was not a regular helix. There were two helices that were not uniformly spaced. And the displacement from uniform spacing could be deduced from the fact that it was the fourth position that had the missing spot. Now, I'm not going to get into the politics of who did what in the discovery of DNA. This has been the subject to a great deal of discussion, and who was venal and unethical and so on. I am not going to give any opinion on that whatsoever. I've simply told you who did what, who saw what, who deduced what, and the basic outcome, the experiment was very simple, and they were working with no money. This was post-World War II Britain, who was broke. Their sample was held on a paperclip. The strand of DNA that Maurice Wilkins had produced was held on an unfolded paperclip, stuck into a piece of cork. The whole thing was stuck into a little chamber that was flooded with hydrogen and flooded with wet hydrogen. We saw the X shape on the consequent diffraction pattern and the way that the X shape then maps into the helical structure of DNA, and the rest, as we might say, the rest of that is history. Okay, so moving on to something else, back to materials. Transformers and beer cans. What do transformers and beer cans have in common? Well, very few engineering materials are used in the form of single crystals. Most of them are polycrystalline, each individual crystal having its own specific orientation, and so we've got a kind of whole jumble of crystals here, which might illustrate the point. When a single crystal gives discrete spots in an X-ray diffraction pattern, a polycrystal, those spots tend to be smeared out into rings. And if the arrangement of those individual tiny crystals in the aggregate is random, the rings are circular and uniform. However, most manufacturing processes give rise to diffraction patterns where those spots are certainly circular, but they're no longer uniform. You get regions that are intense and regions where there's almost nothing. And this indicates that the distribution of crystal orientations is not random. The manufacturing process has in some way aligned the crystals in some preferred way, and we call this crystallographic texture. And materials with crystallographic texture tend to display anisotropic behavior in many of their properties. That means the property is different in different directions. So, transformers are magnetic machines. All centrally generated electricity passes through several transformers between the generating plant and the consumer. Massive transformers at generating plants, and I'm sure everybody has seen these things up wooden poles in the neighborhood. These are so-called pole transformers, and they transform the potential down to 110 volts for domestic applications. The magnetic field of the transformer in the US, a power transformer, reverses itself 120 times each second. Now it turns out that if you look at an iron single crystal, the magnetization behavior of that single crystal depends very strongly on the orientation of the magnetization vector with respect to the crystal axes of the iron crystal. The so-called 100 direction, which I know Dr. Anderson will dislike intensely, which is along the crystal axes, is the direction which is easiest to magnetize and results in the lowest power loss if you have to magnetize and then reverse it 120 times a second. And the direction along the diagonal of the iron's unit cell is the hardest to magnetize and creates the greatest loss of power when you're reversing the magnetic field 120 times each second. And it doesn't matter whether the transformer is loaded or unloaded. This reversal is going on 120 times a second all the time. And so these losses continue all the time. And this is one of the reasons people tell you to pull your iPhone charger out of the wall when you're not using it, because the losses go on even when the iPhone is not on the charger. Anyhow, in 1934, a metallurgist, American metallurgist called Norman Goss, discovered a way to orient the individual crystals in transformer steels by alloying those steels with somewhere between 3 and 4% of silicon, and then following a very complex processing path which involved rolling the steel into sheet, annealing it, rolling it again, annealing it again, annealing it in a controlled atmosphere. Net result of this process was that within that sheet of steel to be used for the electrical transformer, the crystals were very strongly oriented with their easy magnetization directions along the direction in which the magnetic flux was directed in a power transformer. And so this has now gone into production. All the steel companies that produce electrical steels are using variations on Goss's process. The energy losses in the steel cores of power transformers have been reduced from about 10% back in the 1930s and 1940s down to about 1.5% in current power transformers. Now, if you think about it, how much electrical power is generated just in the United States every year? We're now saving 8.5% of that power just by having improvements in the crystal orientation of the steels of which the transformer cores are made. There's a prodigious amount of energy that is being saved. Steel companies are still working on this. I know the research has not stopped. Every year there are incremental improvements in the steels used in transformers. And these losses are slowly and steadily being reduced every year. What does it have in common with beer cans? Well, beverage cans are manufactured by a process called deep drawing. The aluminum for the beverage can is rolled and is stamped into circular blanks. The blanks are placed in a die where a punch comes in, it pushes the aluminum through, the punch comes down, and you end up with a drawn part which is going to form the beverage can. And it turns out that this preferential orientation of crystals in the aluminum alloys as a consequence of the rolling process that produces the aluminum sheet in the first place produces an anisotropy of the ductility of the aluminum sheet, which means it deforms more easily in some directions than it does in other directions. So when the punch comes down, instead of producing a nice uniform circular can, because the deformation is no longer uniform, the end result is a can which has lumps around its rim, which are called ears. This phenomenon of earring results in significant wastage, because to make the can, you have to chop off the entire top of the can where the ears are. And that typically would result in maybe wastage of somewhere around 8 to 10% of the aluminum going into making the can. Now, in the United States, in North America rather, in North America, something like 800 million beverage cans are made every day. I'll repeat that. 800 million beverage cans are made every day. So if you can save 1 cent by reducing the wastage on account of these ears on every beverage can, you're talking about $8 million a day of savings. That's a huge savings and a huge motivation to the aluminum companies like Alcoa and Alcan who make these alloys. It's a huge incentive to do the research to reduce the crystallographic anisotropy in their alloys to improve their product. And they've invested enormous sums of money in doing research to improve their aluminum alloys to reduce the wastage in beverage can productions. I haven't got a slide on this, but the reason I did, Dr. Anderson mentioned I'd worked with Los Alamos and other places. Los Alamos in particular, it turns out this same problem of non-uniform deformation on account of crystallographic anisotropy is a problem when you're trying to compress a plutonium core in a nuclear weapon. If that plutonium core is not crystallographically uniform, it does not compress uniformly. Instead of getting a bang, you get a fizzle out of the bomb. So the nuclear weapons labs have also invested lots and lots of money in processing their product to eliminate these issues of crystallographic anisotropy and, of course, diffraction techniques, X-ray diffraction techniques, the principal tool that is used in doing this research work. And my last topic is going to be electronic materials, the contribution to the development of electronic materials by X-ray diffraction techniques. Start off with silicon, of course, the ubiquitous electronic materials. We're all carrying around silicon in our pockets, in our cell phones. The silicon balls, the silicon ingots that are grown as single crystals and then sliced into wafers. These crystals have to be oriented very accurately because the surface of the wafer, if it is not accurately aligned with the crystal axes of the silicon, when the passivating layer of oxide is put on the surface, develops what are called surface states in the silicon. Now these mess up the electronic properties of the silicon and the chip doesn't work right. And so the manufacturers of silicon wafers have to ensure the correct orientation of their ingots and know what they are before they slice them into wafers. And so X-ray diffraction is typically used to orient the silicon ingot before it's sliced into wafers. The typical X-ray equipment that is used to do this has an accuracy of about 1 minute of arc, or 1/60th of a degree of arc, in orienting the silicon crystal before they're cut into wafers. We're all carrying around quartz in our pockets. We use quartz crystals quartz crystal oscillators to maintain the clocks in our watches, quartz clocks. Quartz crystal oscillators are used in our computers to maintain the clock frequency of the computer, and so on. To maintain frequency stability as the temperature changes, and if you think about it, put your watch on your wrist, you've got to warm up pretty close to body temperature. You take it off at night, put it on your nightstand, it's going to cool down. You want the clock to be keeping good time, day and night. You need the frequency to be stable with respect to temperature changes. In order to achieve that, these quartz crystals have to be cut at a very specific angle to the crystal axis with an accuracy of about 1/10 of a degree, preferably better than that. There are certain angles where if you cut them, the effect of temperature changes on thermal expansion, on stiffness and piezoelectric coefficients all cancel out, and temperature change has very little effect on the frequency at which the quartz crystal oscillator will work. How do they achieve this? Well, they use X-ray diffraction to orient the crystals before they slice them into the little slivers that are put in the quartz crystal oscillators. And my last example is going to be piezoelectric sensors and actuators. We increasingly use piezoelectric materials as sensors, actuators, and transducers. These are used in such diverse things as ultrasonic transducers for medical purposes. They're used by the Navy in sonar arrays, precision actuators that can move a probe over nanometer distances very precisely for such things as atomic force microscopy, and even in lighting our barbecues, we will use piezoelectric materials as gas lighters. A little sliver of lead zirconate titanate here. When I press the trigger, a hammer hits that little tiny sliver of lead zirconate titanate. It generates a potential of about 10,000 volts, creates a spark that lights the gas. And most of you probably have one of these. I always carry one in my pocket. The early piezoelectric materials such as barium titanate have been largely replaced by materials like lead zirconate titanate, known as PZT, as our knowledge of crystal chemistry has improved. This dates back to the work of Pauling in figuring out why it is that crystals containing ions crystallize in certain structures. And as our knowledge of the crystal chemistry of these piezoelectric materials has improved, so the materials that we're using have improved. So barium titanate was an early piezoelectric material used in a lot of applications, but nowadays lead zirconate titanate has mostly replaced it, and there are alloys, ceramics rather, with niobium doping and so on that have even better properties than lead zirconate titanate. Now, I was asked at the end to suggest maybe where crystallography is going in the 20th century. My main prediction is that we're going to have snow. I think it's pretty well guaranteed. And Professor Anderson is rubbing his watch, which I think tells me it's now time that I sat down. So I will sit down and turn the stage over to Professor Moffat.
Right, thank you very much for the invitation to present the talk this evening. As you'll see, it takes a rather different flavor than the fascinating talk that John just provided. So what I'd like to do is to switch gears completely to talk about the scattering of X-rays by much more complicated objects, the objects related to biology. And the discipline here is called structural biology, a phrase that may not be familiar to many of you. So I'd like to talk a little bit about what is structural biology, why is it useful to people in a wide variety of disciplines and industries, and how is it conducted. Since we're celebrating the International Year of Crystallography, I thought it might be appropriate to begin with a video, which, with any luck, will run, which comes from the Royal Institution in London.
Why water boils at 100 degrees and methane at minus 161. Why blood is red and grass is green. Why diamond is hard and wax soft. Why glaciers flow and iron gets hard when you hammer it. The answers to all these problems have come from structural analysis. That's how the Nobel Prize winner Max Perutz summed up X-ray crystallography. Never heard of it? Don't worry, not many people have. Yet it's arguably one of the greatest innovations of the 20th century. 28 Nobel Prizes have been awarded to projects related to crystallography, and the very first of those is where it all began. It's now 100 years since, following early work by Max von Laue, the first structures were determined by father and son team William and Lawrence Bragg. In 1913, they fired a narrow beam of X-rays at a humble salt crystal and photographed the diffraction pattern as the crystals split the beam into many rays. Lawrence soon realized that this pattern held the clues to the atomic structure of the crystal itself. The equation he developed, Bragg's law, made it possible to work out how the spots in the diffraction pattern are related to the specific arrangement of atoms in the crystal. 2 years on, and the Braggs were awarded the Nobel Prize. Impressive stuff. Not only that, the Braggs mentored a dream team of crystallographers who went on to work out the structures of a huge range of molecules. From Kathleen Lonsdale, J.D. Bernal, and Dorothy Hodgkin, to David Phillips, John Kendrew, and Max Perutz. Remember him? Plus, Rosalind Franklin and others even helped map the structure of DNA, probably the most famous result of X-ray crystallography. Today, the work hasn't stopped. Crystallography remains the foremost technique for working out the atomic structures of almost anything, which is very useful for finding out why things behave the way they do. From the metallic structure of the blades of a jet turbine to the immune system fighting off viruses. Turns out, Max was right. Modern crystallographers are doing exactly the same thing as the Braggs, just at a larger scale with more sophisticated mathematical methods and more impressive machines. Crystallography is even reaching beyond our planet. The Curiosity rover is now performing X-ray diffraction analysis of the soil on Mars. But there's plenty of unfinished business back on Earth. There are still many thousands of complex molecules to look at, and a lot more questions to answer.
Doesn't seem to want to advance. That's a slight problem. Let's try. There we go. Right, thank you. Okay, so structural biology, discipline of which crystallography is a part, can be thought of as the study of anatomy at the atomic, molecular, and cellular level, and the very powerful role of crystallography in biology is that it reveals this anatomy at the atomic and molecular level. So we're using structural biology to understand biological function. That is how molecules and cells work, and indeed how that working can be interfered with by drugs, for example. So the relationship between structure and function at the biological level could be thought of as analogous to that between anatomy and physiology at the level of the whole organism. And there's a famous quote from Francis Crick: if you can't understand function, you'd better study structure. And a little aside here, John told us about the pioneering studies of the structure of DNA by Rosalind Franklin, Maurice Wilkins, and then the interpretation by Francis Crick and Jim Watson in Cambridge. What he didn't say was that Francis Crick at the time was not actually studying DNA as a research student. He was a research student in X-ray crystallography of hemoglobin with Max Perutz. And if you look at Francis Crick's PhD thesis, which I've done, it's entirely about boring crystallography, these early days of hemoglobin. The structure of DNA is nowhere mentioned in the text, and a reprint of the paper is not bound in or inserted in a folder at the end of the thesis. So for Crick, the structure of DNA was really a sideline on his PhD thesis. We should all be so lucky, all right? But in any case, we're studying structure in structural biology in order, I would assert, to understand how molecules work. Now we can use X-ray scattering to image objects, molecules, cells, and higher entities on structural length scales ranging from the atomic, such as those we saw in John's talk, to the cellular. But there's a problem here. Cells are indeed the basis of life. So here on the left-hand side, we have a cartoon of an intact cell, and it's an extremely complicated structural entity. Within it, in the nucleus, for example, we have the DNA involved in storage of genetic information. The information in that DNA is processed or transcribed, as we would say, by an enzyme called RNA polymerase, based on, and then generates messenger RNA, mRNA. That is subsequently translated by a machine called a ribosome and made into protein. The cell depends on energy from ATP synthesis, and perhaps the cells may be infected by viruses. So cells are the basis of life, but cells are not structurally reproducible. This is simply a cartoon of a cell. As we shall see in a moment, if I show you an image, an X-ray image of a real cell, it happens to be a yeast cell in the middle here. If I were then to show you another image of another yeast cell from the same yeast in this case, it would look different. It would have the same constituents, nucleus, mitochondria, vacuoles, nucleolus, etc. But it would not be spatially reproducible. So although cells are the basis of life, you cannot actually crystallize cells, because the nature of crystals, as we've already seen, is that the entities being crystallized, whether they're atoms or small molecules like sucrose, have to be spatially reproducible. Cells are not. So in fact, what we are trying to do then in structural biology of cells is imaging these non-crystallizable entities by various forms of X-ray microscopy, like the one we just saw. So our structural biologist's dream then is to determine and understand three-dimensional structures of all the individual macromolecules together with their complexes and assemblies within a cell. The individual macromolecules, hemoglobin, for example, or ATP synthase, or an enzyme like RNA polymerase, are structurally reproducible, and therefore they can be crystallized, and we can use the techniques, the very powerful techniques of X-ray crystallography. So more specifically, we're asking, how do biological macromolecules actually fold up and assume this definite three-dimensional structure? What is their anatomy at the molecular level? How do they assemble into more complicated structures? I'll show illustrations of those in a little bit. How do they work? How do they function? What do they do? And how are they regulated or controlled? How do they change their structure as they work? I'll address that last question at the very end of my talk. And ultimately, we're asking, how are cells formed and how do they function? Cells, then, are the basis of life, but the individual molecules within them are susceptible to X-ray crystallographic approaches. Here's another illustration, a real experimental illustration now of just how complicated a piece of a cell is here, shown on the in this experimental image here. This is in fact obtained by three-dimensional electron tomography. It's rather analogous to the tomographic images of a turbine blade, or I guess it was being drawn aluminum that John illustrated. And here, individual types of molecules are color-coded differently in this graphic representation. Actin is sort of brownish things, look like pretzels, actin filaments. Ribosomes are these green blobs. We'll see more of them in more detail a little later. And then there's a small section of membranes here. Here's one up here on the left-hand side, and other little bits and pieces down here. And look at the scale of this. We're down at the scale of hundreds of nanometers here. This happens to be obtained by electron tomography, but you can also form images increasingly by X-ray tomography. So this is not crystallography, but it's another way of using X-rays to explore structure. So X-rays, as we have seen essentially the whole evening, provide a very powerful method of determining structure at the atomic level. Crystallography, that I'll concentrate on now, and also imaging. Crystallography of individual, spatially reproducible molecules and their complexes, and then imaging of non-crystallizable entities, intact cells, the components known as organelles at the subcellular level within cells, or even indeed of whole organisms. So here's an example of using X-rays at the Advanced Photon Source at Argonne to examine, in fact, an entire organism. So this is an X-ray image rather analogous to that of the one that a dentist might take of your teeth, and of this little insect here. And the physiological problem here the investigators were pursuing was exactly how the insect trachea expands and contracts as the insect is in fact breathing. So here is X-rays being used as an explicit form of imaging, again, of a non-crystallizable object, in this case, an intact insect. So if you're going to carry out X-ray crystallography or imaging, you need a suitable source of X-rays. And when crystallography began, and John gave various illustrations of this with the Braggs and von Laue himself, the X-ray source was lab or even office-sized. So here, in fact, is an image from 1925 of Linus Pauling's X-ray equipment in his office at Caltech. So the X-ray equipment itself is housed in this box here. I don't know what it was made of. I hope it was made of lead. The X-rays are coming out of a little tube here. It's a little hard to see. But here is his desk. And here are an array of scientific journals, lab books, or what have you. And here is his safety equipment, his Homburg hat. So this is really, well, it's small scale. It's sitting in his office, but it's also scary by modern safety standards to have the equipment with X-rays, which are hazardous, in your office and not very well shielded, and you're sitting only a few feet away, and your safety equipment is a conventional hat. Nowadays, we've come a long way in our X-ray sources. So now the X-ray sources that almost everybody uses are called synchrotron X-ray sources. These are not office-sized, they are building-sized, and here's an aerial view of one such source, the Advanced Photon Source at Argonne National Lab. And it's this very large donut-shaped building which houses within it a vacuum chamber, also donut-shaped. In the vacuum chamber is embedded in an array of magnets, and as the electrons circulating in this vacuum chamber are accelerated by passing through these magnets, they emit electromagnetic radiation, and we arrange things in a way that they emit X-rays that are useful for either X-ray imaging or X-ray crystallography. Now this is a huge machine, so one can conduct roughly 100 X-ray experiments simultaneously, not just one experiment at a time as the Braggs or Pauling did. It serves somewhat more than 4,000 experimenters per year in a very wide range of scientific disciplines. We've already seen that X-ray techniques are widely applicable in a wide range of disciplines. And however, and this source also is roughly a million times brighter than Pauling's office-sized X-ray source. These are extremely intense X-ray sources. So what does the raw data of X-ray scattering from a crystal look like? We've seen various cartoons, but I thought it might be useful to show some real data. So here is a modern version of a Laue diffraction pattern. We heard about von Laue in John's talk. in which a stationary crystal is illuminated by a beam of X-rays that contain many different X-ray wavelengths. So the X-ray beam, to record a pattern like this, would have been coming more or less perpendicular to the plane of the slide. The crystal might be located about a meter or so in front of the screen here, and the X-rays would be scattered according to Bragg's law, and would give rise to this array of spots that you see here, slightly lighter in color, on this grayish background. It's a little hard to see here, but in fact these spots extend to the very edge of your X-ray detector here. We can just trace of it down the bottom. And there are about 4,000 usable reflections or spots on one image like this. So this is a real experimental image. It happens to come from a crystal of myoglobin, which John Kendrew famously studied and solved the structure of the first atomic structure of a biological macromolecule. The raw information in these X-ray scattering patterns resides in the location of these spots, and that depends on the size of the building blocks in the crystal, and on the intensity, in this case the whiteness, of each of those spots. And unfortunately, it also resides in another quantity that you cannot measure directly, which is called the phase of each of these spots. And in fact, determining that phase is the main business of crystallographers. You cannot measure it directly. It's non-trivial to obtain. Were that not the case, crystallography will be a completely straightforward discipline, but it isn't. So here's what the raw data looks like. You can crystallize even bigger things. Here is a Laue diffraction pattern from a virus crystal. It's a crystal of a plant virus, bean pod, bottle virus. And this pattern was obtained in about 30 seconds. What I did not say is that this pattern here was obtained in about 100 picoseconds, 10 to the minus 10 seconds, from a synchrotron X-ray source. And this was also a synchrotron X-ray source. So it is possible to crystallize biological entities as complex as certain viruses, and they give these wonderful X-ray diffraction patterns. So X-ray crystallographers in structural biology have been pushing ahead with solving yet more and more complicated structures. Highly complex molecular machines, arrays of molecules, lie very much at the frontier of structural biology today, and they are determined using X-ray beam lines at the Advanced Photon Source, and let me illustrate those with two such results on the ribosome and what are called G-protein coupled receptors, or GPCRs. We've already heard a lot about Nobel Prizes, and these two structural studies at the APS were indeed recognized in this way. The 2009 Nobel Prize in Chemistry went to Venki Ramakrishnan, Tom Stites, and Ada Yona for their independent work determining the structure of the ribosome. So here is the result of a crystallographic structure determination. And these ribosomes are extremely large and complex machines, molecular machines. They contain many RNA, ribonucleic acid molecules, and many proteins. They have an extremely large number of atoms per crystallographic building block or asymmetric unit. And they have very large building blocks. The largest building block is about 2,700 angstroms, 270 nanometers. Determination of a structure as complicated as this required these very highly parallel, very intense X-ray beams, and a particular form of those were developed at the Structural Biology Center at the Advanced Photon Source at Argonne. To state the obvious, to determine the crystal structure of the ribosome, you need crystals, and that is where the major contribution contributed by Ada Yona, who's an Israeli scientist, who first succeeded in the face. Everyone else thought she was crazy to even attempt this, but she first succeeded in crystallizing ribosomes that were obtained from a salt-loving bacterium found in the Dead Sea in Israel. So here is an example then of today's frontier in structural biology, and here's another one. The 2012 Nobel Prize in Chemistry awarded to it, shared by Bob Lefkowitz from Duke and Brian Kabilka, an X-ray crystallographer from Stanford University. So why should we be concerned about the structure or interested in the structure of these G-protein coupled receptors? Here is an image of one such protein molecule here in the bottom right. The different proteins here, making up this complex, have different color representations. Again, an extremely complicated molecular entity. Well, about 75% of the drugs on the market today interact with membrane proteins such as these GPCRs. So if you were a pharmaceutical chemist, for example, you are intensely interested in molecules such as this. If you understand the anatomy of your target, GPCR, at the molecular level, then the argument goes that you will be able to design better drugs to interfere with the working of this molecular machine. It's as though you come down from Mars, you look at a car engine and try to figure out how it's working. You can just study it when it's not running. You could probably eventually figure out how it worked, but that's a bit difficult. Better yet, to start it running. And then if you observe it running, you could figure out, well, maybe if I pull off some of these leads on the top, the spark plug wires, it'll not work as well. If I pull them all off, the engine will stop. And likewise, if I eliminate the fuel, the engine will stop as well. The same sort of reasoning goes into trying to figure out how to perturb the function of complicated molecular machines like these G-protein coupled receptors. These scientists were able to grow very tiny crystals, but only very tiny crystals, of these G-protein coupled receptors. Those interact very weakly with the X-rays. They scatter X-rays very weakly. And since the crystals are very tiny, you need very tiny but very intense X-ray beams. And these approaches were pioneered at a European synchrotron facility, and there were significant further developments of this technology at the Advanced Photon Source, the GMCA CAT, and key data on these microcrystals was also collected at the Advanced Photon Source. So these GPCRs, as I said, are extremely important transmembrane proteins. They are the chemoreceptors that transmit chemical signals across the cell membrane, from the outside of the cell membrane to the inside. And as I said, they're the target of many therapeutic drugs, such as, for example, beta blockers. Here on the left is a photomicrograph, a light photomicrograph of these tiny crystals of these GPCRs. So here's one, it's another here. Notice they have sort of ragged ends, sort of like little snowflakes here. And these blue ellipses here, or little circles, represent the X-ray beams, the size of the X-ray beams used originally at the APS and on the synchrotrons to explore these crystals. So this particular X-ray beam size is 100 by 30 microns. But these microbeams, more suitable for looking at these tiny crystals, are only 5 microns in diameter. They're represented here by these little 8 traces of X-ray beam here. The X-ray beam is going to scan across the crystal, and here we have shown it in 8 different positions. And the atomic structure of these GPCR is shown here, embedded in a membrane. The part of the membrane is shown by these lipids here. They're in white and red, with phosphate head groups in red. And here is the molecule itself, again, extremely complicated, embedded in the membrane. The orange objects here are cholesterol molecules, which are also all part of the membrane. So the use of these synchrotron X-ray sources, such as the Advanced Photon Source, was absolutely essential to this Nobel quality research. The scientific teams that use the facilities are large, they are international, and they're often multidisciplinary. Notice that although the ribosome and the GPCR structures that I illustrated attacked biological and biomedical goals, the Nobel Prizes were awarded in chemistry, not in physiology or medicine. And this is actually an illustration of the interdependence of the sciences, and in particular, in the role of fundamental science in today's biomedicine. We're sitting here in a medical science building at Northwestern University, but you can't believe it, but the researchers in this building and those in corresponding buildings at other medical schools around the country, and on the south side of Chicago at the university, of Chicago, are highly dependent on advances in basic physics and chemistry. And these two structures here illustrate that. I should also point out that both the ribosome and these GPCR projects, although conducted within a university environment, have spun off new companies aimed at the development of more effective drugs based on the knowledge of the three-dimensional crystal structures of, for example, bacterial ribosomes, which differ from human ribosomes. So if you could interfere with the bacterial ribosome working, but not with the working of the human ribosome, you can and do kill bacteria while preserving you and not giving you nasty side effects like a bad headache. But these illustrations and those that John showed earlier are all static structures. How about dynamics? Well, all reactions in biology and in chemistry involve structural change. Atoms, or the electrons that are interacting with the X-rays, have to move around for a reaction to occur. So can these structural changes be visualized? Can we turn crystallography from the static structure that it has been essentially since it was founded into a dynamic experimental technique? That I'll end with this. It happens to be my own particular area of interest. So if one is going to study dynamics, you need new technology to do it. And this notion of devising new technology to look at a new scientific problem is, of course, just about, goes back to the dawn of science. And let's illustrate this from a century ago. The American physiologist Edward Muybridge, 1878, devised the first movie camera in order to address the biological problem of what formed the gait of a horse take. So here we have the individual frames of the movie that he obtained famously with his first movie camera. And he was able to ascertain, in fact, that there were, here's one illustrated here, and all four hoofs of the horse were off the ground at the same time. So devising new technology, in this case, to look at dynamics on a length scale of centimeters or meters and a time scale of seconds, as the horse was, in fact, I think it's cantering down the track. So we're going to do dynamics, but we're going to do it not on horses, we're going to do it on individual molecules. And in fact, the molecules in which we and others are interested are light-sensitive molecules. The one we work on a lot is called photoactive yellow protein. And here is the tip of a crystal of photoactive yellow protein here. This little blue box here that you may be able to see illustrates a portion of this crystal that we're going to illuminate with X-rays. And that box is 40 by 80 microns. And the entire photoactive yellow protein crystal here, of which this is just the tip, is about 100 microns across, say, over here, and about 1,500 microns, 1.5 millimeters long. It's extending a long way. In fact, it's extending out into the hole behind us here. So it is possible to grow sometimes quite large crystals of biological molecules. So this protein is a blue light photoreceptor. It's found in bacteria, and bacteria like to sense the light environment in which they are located. They don't have eyes as such, but they do have photoreceptor molecules that can sense the quality of the light that's falling on them. They can sense the wavelength of the light, they can sense the direction from which it's coming, and sometimes they can sense its polarization. So we're interested in how this process of signaling by these photoreceptors actually work. When the bacterium senses light of what it regards as the biologically appropriate quality, it will swim towards that light. If it senses light of inappropriate quality, too intense, wrong color, something of that sort, it will swim away from it. So how does the bacterium know in which direction to swim? So here in the center panel B here, we have the crystal structure, the static crystal structure of the photoreceptor. It's a blue light photoreceptor. Most protein molecules are colorless. So if you're going to absorb blue light, you need a non-protein part, and that non-protein part here is shown in this box, dash box in the center here, and blown up on the left-hand side in panel A. And that non-protein part is a small organic molecule called para-coumaric acid, PCA, and here it is blown up. When this non-protein part here, the chromophore PCA, absorbs light, two things happen. First of all, it changes its chemical structure. This tail of this molecule here that connects the head of the molecule to the rest of the protein down here contains what chemists would call a double bond. And the geometry of atoms around that double bond changes in response to light. In the dark, no light. The disposition about this double bond between C3 and C2, labeled here, these two atoms, is, the chemists would say, trans. If it absorbs a photon of light, isomerization occurs, and the disposition about this bond changes radically and becomes what the chemists would call cis. So a molecular convulsion has to occur on absorbing light, but it's trying to do that within a very crowded molecular environment, the interior of the protein. This chromophore, although it's not obvious from the central cartoon, is in fact completely buried. And so, how is it going to execute this molecular convulsion known as isomerization? When it's buried and banging into all these other atoms, its protein neighbors. So that's one thing we'd like to understand. Secondly, and this is shown in panel C, the protein changes its color as or after it absorbs a photon. It starts off in the dark in this ground state here labeled PG, absorbs light, that's what H nu means, goes to an excited state, which then decays in a series of intermediates. That's the right-hand side of this photocycle or clock face here. And these various intermediates, shown here in brackets or no brackets, have different colors. In a few picoseconds, very fast indeed, we get intermediate we label I0 in about 100 picoseconds, another one, nanoseconds, yet another one, a few hundred microseconds, another one, and eventually you regain the ground state in a time scale of hundreds of milliseconds. So this is a resetting system. It absorbs light, and then after 150 milliseconds, a second or so, it returns to the ground state, absorbs more light, and so on and so forth. So how is this actually working at the atomic level? So we have devised and applied techniques of time-resolved X-ray crystallography to watch the dynamics of this system. And here I'm about to show a movie. We got a face-on view of the chromophore here. Perhaps we could dim the lights here. The individual atoms are shown by little black dots here, and the bonds between them, these thin lines. In the bottom left-hand corner of either the face-on view or the edge-on view of the chromophore here, we see a little clock face. It's going to be running by time, so this is going to progressively advance around the clock face as time proceeds on a time scale from 100 picoseconds to nanoseconds. So these structural changes that we're about to see are happening extremely fast. Notice the image as we start off here is entirely black and white. You're going to see magenta and green features appear flanking these atoms. And these magenta and green features, they're actually a representation of raw experimental data. And they will illustrate that atoms, these individual atoms, or more precisely the electrons within these atoms, are moving as isomerization is occurring about this double bond here. Notice it's about halfway across the image and about a third of the way up here. So big changes are going to occur here. So now let me run this movie. With a line work before. Yes. So here we have the face-on view and the end-on view, and here is our clock running here. So you see already after 100 picoseconds, lots of magenta, lots of green. Atoms are moving fast. And these colors will evolve as time is running here on our clock face. So this is the raw data of time-resolved X-ray crystallography of the elementary chemical process of isomerization in this particular system. I'll just show it once, but I'll be happy to show it afterwards to anyone who wants to take a look. We can interpret those results as well. So here is an interpretation. Again, we start off with our chromophore. Here we're seeing it in a more detailed atomic representation here. Isomerization is going to occur about this bond here, and we will see the movie. This is an interpretation of the data I just presented as the molecules. Whoops, sorry, wrong one. Let me do that again. Here we go. So here isomerization is occurring. This oxygen atom shown here in red is moving around. It started off to the left. It's going to go over to the right. And the entire chromophore buried in the interior of the protein is going to undergo this exquisitely choreographed set of atomic motions. When each symbol flashes here, that's an experimental result. We are interpolating between these experimental results based on computational calculations of the electronic structure of what's going on in this chromophore. This is known as a bicycle pedal pathway for isomerization. There's another pathway this chromophore also exhibits called hula twist. It should be no surprise that pathway was proposed by a physical organic chemist from the University of Hawaii. Okay, so finally then, I would assert that research in structural biology today cannot be effectively conducted without ready access to sophisticated, large-scale, and yes, I have to say expensive user facilities such as these X-ray facilities afforded by the Advanced Photon Source at Argonne and similar facilities around the world. Secondly, I think it's fairly evident from both our talks that scientific advances in X-ray crystallography depend on a century of progress. The sources, the experimental techniques, the algorithms for analyzing data, the computing power to execute the calculations, and the molecular graphics for displaying the final results. When John and I were students, the word computer did not refer to a server, piece of hardware. It referred to a job description. Computers, when I was a research student, were attractive young ladies who lived in a small office down the hall and worked on mechanical calculator devices to carry out some very complicated arithmetical sequences. So we've come a very long way, and our ability in structural biology to determine complicated and, I would say, useful entities like the structure of a ribosome or these G protein coupled receptors, our ability to do that is dependent on scientific advances in X-ray crystallography and X-ray imaging in a very wide variety of areas. But if you have a big facility like the Advanced Photon Source, you can't stay still. So there are plans to upgrade the facility, and the general goal is to determine structure and dynamics in biology from proteins to intact organisms. So here we have proteins, and here we have an intact organism, and the length scale of these entities is varying, of course, very directly. And here are various scientific jargon words for the various upgraded, a wide variety of X-ray-based techniques that we are planning at the moment, and we'll implement in 5 years or so. I already talked about tiny crystals. The ability to look at them was essential for the GPCR project. I ended with studies of fast dynamics on the picosecond time scale, and there, I illustrated much earlier, wide field imaging of larger objects such as intact insects, and these other techniques I haven't had a chance to talk about. So X-rays are alive and well, and as a discipline, we still have lots of fun using them. So with that, thank you very much.
Thank you, Dr. Moffat. That was a wonderful demonstration of the convergence that's necessary in many fields of science and engineering, I would say, to make things work. And it's true in most fields that nothing is in isolation. It's always built on what they call the shoulders of giants, and Newton said, but built over time with advances. And so we can't just look at one advance and say, well, that was due to one line of funding. The funding has come to other fields. It's getting a little late, but there's my time here, I think, for 1 or 2 questions. Does anybody have a question? Preferably a non-expert the experts can ask questions then.
Is there an advantage to cooling static samples, or is the process so fast that you're not going to see any kind of motion?
So
The static crystal that you're looking at.
Okay, so the question was, is there an advantage in cooling the samples to perhaps slow things down or make them somewhat easier to see? Yes, the rates of chemical reactions are temperature dependent, but what I did not say is that most crystal structures, static crystal structures these days, are determined at near liquid nitrogen temperature, around 100 Kelvin, and at such temperatures, atoms by and large do not move. The molecules are literally dead, they are inactive. And so if you want to do dynamics, you cannot overcool, if you will, your sample. What that means in practical terms is the environment of the molecules within these crystals still has to be fluid. So that really means that you're restricted to nothing much lower than your average Chicago winter temperature. The gentleman up there.
Okay, yeah, my question is also regarding samples. Can you talk a bit about how you prepare them and how they look before you finish them with X-ray?
Please repeat the question.
Yes, so the question was, could I talk about how the crystals are actually prepared and what they look like, etc. Okay. What they look like is the easy part. They look just like crystals of common salt or snowflakes or what have you. From the external form of the crystal, you cannot tell what the molecular constituents are. Okay. How do we prepare them in the biological case? Well, I emphasized at the beginning of my talk that if you're going to crystallize anything, they have to be structurally reproducible. They have to look the same. And John also told us at considerable length that if you've got an alloy, for example, it's obviously not homogeneous. A copper atom doesn't look like or bond like an aluminum atom, for example. So if I want to grow a crystal of a biological macromolecule, I have to purify it to make sure it contains only the desired GPCR and nothing else. But these molecules, GPCRs, are isolated from a cellular environment which is very heterogeneous. There's lots of different types of molecules there. So first step is elaborate purification approaches. Okay? And there are ways of testing how pure, how chemically pure your entity is. Then the general process of getting these biological crystals is to adjust the solution conditions so that they very slowly become insoluble. You hope they form an ordered crystal and not an amorphous or disordered aggregate. But sometimes you're lucky, most of the time you're not. So it would be fair to say that growing protein crystals is still something of a black art. We know what the techniques to use and how to go about it, but we cannot offer any guarantees of success of a given entity. Therefore, we try many hundreds, sometimes thousands of different solution conditions to approach insolubility, and you keep your fingers crossed that indeed you will get crystals and not nasty, disordered precipitate.
So I actually had a question for the first speaker. I didn't bring my umbrella today. But we were talking about pure metals and how they were so soft because they have the dissolutions in them. So I was wondering if it would be theoretically possible to make a really hard pure metal if there was just a way of making them perfect in such things.
Yeah, the question related to the fact that pure metals are very soft on account of the presence of defects. And whether or not it's possible to eliminate those defects and thereby make the metal hard and strong. Yes, it is. It has been done in the lab. Very tiny crystals called whiskers have been made, which typically do contain one dislocation running down the axis of the whisker. It's called a whisker for obvious reasons. Okay? But that dislocation is basically harmless with respect to the properties of that whisker. And they have been shown to be essentially the theoretical strength that the bonding would predict. On the other hand, it's rather difficult to make anything product out of such research. Some years ago, when I was a postdoc, I visited the University of Oxford. Forgive me for saying such a bad word. But I went to the University of Oxford to meet some of the faculty there, and they had been growing macroscopic-sized crystals of copper. When I say macroscopic, I'm talking maybe 2 centimeters long, centimeter in diameter, with very complicated processes to grow, very, very slow processes to eliminate the presence of dislocations. And they indeed also, on testing, very careful testing, showed incredibly high strengths, just like the theoretical strength you might expect. But the disadvantage is, first off, it was incredibly slow, so there's no way you could ever make a commercial product that way. And secondly, if you accidentally dropped it, oops, it filled with dislocations and became normal. And so it was really a lab curiosity and nothing beyond a lab curiosity. To the best of my knowledge, this is theoretically interesting, but there is no active research, to my knowledge, going on in any way suggesting that this might ever, in the near future at any rate, this might be commercially possible. Now, I'm not in the business of predicting the future any more than Niels Bohr was. I'm not going to say it's never going to happen, but right now, I don't think it's a serious research endeavor to try to do this.
So, here and here.
Were there any like advantages of that pure small crystal color that they could fall?
I don't think that was pursued, but the presence of these dislocations, this is a very interesting topic in materials science. It's been going on for the last 70 years. Is what effect of these defects, and dislocations are just one type of defect, what effect do they have on the properties of materials? And they have, in general, a deleterious effect on electrical properties too. They increase the electrical resistivity or reduce the conductivity of copper or aluminum as used in wiring. And so the wiring is used to bring power to our homes, from the generating plants down at the nuclear power plants, 50, 60, 70 miles from here, basically coming along aluminum cables. These are high purity, carefully processed, annealed aluminum. They're very soft in order to give them strength so they don't come down in a wind or in a tornado. They have to be sheathed in steel wire to provide strength. So when you look up at those power lines you see along the side of the freeway, as you drive down Interstate 80, for example, you go miles and miles and miles, you'll see these overhead power lines strung along. They're composite cables. They have a very high purity, highly annealed, as defect-free as they can make them aluminum core, and they're surrounded with a high-strength steel sheath. The steel provides strength so they don't sag under their own weight, and the aluminum provides the conductivity. Essentially, maybe 5% of the current flows through the steel, and 5% of the strength comes from the aluminum. Okay? And no one has yet figured out a way of making a homogeneous cable that has both strength and high electrical conductivity.
Because the metals have a tendency to be soft because of their crystallography, would it be a tendency now to use more carbon composites for commercial products rather than metal alloys?
Yeah, the question related to the issues with metals and whether carbon composites, if I could paraphrase, can be used to replace these. The answer is yes. There are a lot of applications now where carbon composites are being used for exactly that reason. Carbon composites are very stiff, they're very light, they're very strong. At the moment they're rather expensive. If money is no object, like you want them for your golf clubs or your tennis racket, or you don't care how much you pay as long as you beat the next guy. Well, you'll see a lot of carbon composites in tennis rackets and golf clubs. You'll see carbon composites in camera cases, in crash helmets. some aircraft components, but although carbon composites have been in development ever since I was an undergraduate, and probably before, there are still issues particularly concerning toughness and resistance to fracture, which is why you haven't seen airliners with carbon composite wings yet. There's a lot of issue concern with certifying structure.
Dreamliner had those.
Dreamliner has a lot of carbon components in it, but essentially not in the wings, not in the really serious structural components, not in the fuselage. But there was an Airbus crash some 10 years ago in Queens, Long Island. And that was due to essentially a failure of the tail of that Airbus. And there were some issues, it was kind of pilot error. But what happened was the tail, which was a carbon composite, hadn't been designed with a certain pilot input in mind. When the pilot rapidly moved the rudder pedals, the tail came off and the airliner crashed in Queens. You may remember that. It's about 10, 12 years ago.
Dr. Goldman, one last question.
Okay, so in the current facilities today, like APS, we use very high energy X-rays. And the question is, to what extent is the process limited by the energy of the X-rays disrupting the molecular bonds? I would assume it's more so in biology than non-biological molecules, but it has to do with the bonding strength and so on. So are there restrictions or limitations of what you can do because of that?
So the question has to do with exactly how X-rays are interacting with matter, and are there damage effects that restrict what you can do with these very intense X-ray sources like the Advanced Photon Source? The answer is yes. So what's going on with when X-rays interact with matter is some of the X-rays bounce off, or as we would say, elastically scatter from the electrons and give rise to these wonderful X-ray diffraction patterns. We like that. We can use that elastic X-ray scattering. However, X-rays are also absorbed by atoms and electrons, and you then get inelastic processes, all of which deposit energy in the sample, and which, as you're alluding to, tend to rip the molecules apart. However, ripping the molecules apart takes time. It does not happen absolutely instantaneously. And in a brand new type of X-ray source, which I didn't talk about today, called a hard X-ray free electron laser, it's actually a laser source, this process of damage is roughly 10,000 million times higher than it is even at the Advanced Photon Source. So one is critically worried about damage. Okay. These X-ray laser beams are sufficiently intense that they will fully ionize all atoms in the sample. They will strip all electrons of all atoms. So that doesn't sound like a very good idea if you want to study the structure of the object you're illuminating. However, it takes about 100 million, a couple hundred femtoseconds for this to happen. So it turns out you can collect your X-ray diffraction signals, such as the ones I showed here, on very, very tiny samples of even biological crystals, which are indeed radiation sensitive, and collect them within 100 or a few tens of femtoseconds. Okay? So you can outrun radiation damage with time. And in fact, that's basically the only real reason that these X-ray free electron lasers are going to be useful. If everything you looked at was instantaneously damaged, then you could not obtain the beautiful structural information that John and I have been talking about this evening, but there is at least one way around it. So the free electron lasers and what you can do with them probably would have to be the subject of a future lecture to this group.
Thank you. That was a terrific, these two terrific lectures. I guess the greatest compliment I could make is, if I had had either of you gentlemen when I was taking crystallography, I probably would have studied a lot harder and learned a lot more. Great, thank you very much. Remember that if you're not a member of C2ST, please sign up. Peter, would you stand up if This gentleman will take your membership. It's very affordable. You'll feel great after you sign up and come to lectures like this, and you'll advance the cause of the Chicago Council of Science and Technology. Thank you for being here.